We are excited to have Dr. Barry O'Keefe present our next topic. Dr. O'Keefe received a Bachelor of Science degree in botany from Michigan State University and a PhD in pharmacognosy from the University of Illinois at Chicago. At the NCI, Dr. O'Keefe currently leads the Protein Chemistry and Molecular Biology section and is Deputy Chief of the Molecular Targets Laboratory. He is also Chief of the Natural Products Branch, which is responsible for the collection, extraction, prefractionation, and discovery of bioactive natural products. I'm confident you're going to enjoy today's lecture.
Hello, my name is Barry O'Keefe. I'm the Chief of the Natural Products Branch here at the National Cancer Institute. And what I've come to talk to you about today is natural products research. A lot of you might be familiar with natural products. A majority of the population of the world relies on plants and microbes and a variety of other natural products for their health needs. The NCI, in fact, has had collections of natural products taking place for over 30 years and has amassed an amazing repository of biodiversity, which the NCI uses to test for compounds that might be useful in cancer treatment. What I'd like to talk to you about today is the aspects of the NCI involved with screening natural product extracts to identify modulators of targets and pathways in cancer. So with that, I'll move along to my presentation. So to start out, the outline for today's presentation, we'll discuss why we even screen natural products in the first place, and then we'll discuss some of the challenges associated with screening natural products. And there are a variety of those, and they entail the necessity of making a variety of screening decisions on how you intend to use your natural products in your development of screens to be able to identify new molecules. I'll then discuss a couple examples of cell-free screens that we've performed in our laboratory, and then we'll conclude with some new avenues for improving the efficiency and increasing the output of natural products research. So, why investigate natural products? Well, first, what you're seeing here is a graph that shows the year-to-year new drugs that are approved by the FDA. And this shows in the green bars the percent of those new drugs that are derived from natural products. So as you can see, there's a significant proportion of all drugs that are natural product derived. In this case, about 30% of all small molecule drugs approved by the FDA over time have been natural product chemotypes. And for anti-cancer in particular, greater than 40% of all anti-cancer drugs are derived from natural products. So this is a very fruitful area of research for the discovery of new natural products. So just to give you a more pictorial example, here I'm showing a variety of the unique chemical structures that are found in natural organisms that have been approved drugs. These are just those that were approved from 2007 to 2012. And what you see here are 11 different compounds that have been approved for either cancer, or in one case we have one here that was approved as an antibiotic. And you can see there's a lot of structural complexity and a lot of difference to the structures here. And that's based on the fact that some are from plants, some are from microbes, some are marine organisms. What is most fascinating to me, however, being involved in natural product discovery, is that even though we've had this level of success with natural product research, less than 1% of the total biome of potential organisms for natural product research have been examined. So there's still a lot of work to do. So, if natural products are so successful and they're responsible for so much of the drug pharmacopoeia that we use on a daily basis, one of the things that's most vexing to us natural products researchers is the fact that recently, over the last decade or so, high throughput screening has been development in drug discovery. So these are large libraries that are screened in a very high throughput to identify molecules that could be useful drugs. And what I show on this slide is a graph that was put together by my colleagues. And what you see here in the white bars are the number of papers in PubMed that show an increasing amount of high throughput screening, or HTS. So these are all the citations for high throughput screening on a year-to-year basis. What you see in red, barely visible down at the bottom, are those screens that actually involve natural product extracts. So even though natural products have been responsible for such a high percentage of the drugs, the amount of modern drug discovery screening that actually uses natural products is only a very tiny percentage, shown by those little red bars there at the bottom of this graph. So the question is, how do we get more people screening natural products and get them involved in this area of research? Well, one way is the NCI has a natural products branch, which is responsible for collecting source organisms from around the globe and growing microbial source organisms. We also then process and extract these organisms to produce aqueous and organic extracts that are then provided to researchers worldwide, both in vials and as screening plates. We also are involved in the isolation and identification of active compounds from these extracts, and also we can work with people to re-isolate bulk quantities of the compounds that are isolated so that there are more compounds available for animal studies and preclinical development. We also are responsible for large data fields of where these organisms were collected, the GPS coordinates, what the taxonomy is. We also have agreements with the countries that allow us to collect these organisms. There are letters of collection that are in fact almost a treaty between the United States and the host countries that ensure that the host countries' rights are respected. So should anything come that is commercially developed out of one of these organisms, there are responsibilities for those developing that and licensing that they have equitable benefit sharing with the host country. And we think that's very important. So where are collections located? Here you see a world map, and you can see different hexagons on blue, green, red, and also black, and these show the locations of the collections of the Natural Products Branch around the globe. You can see the majority of them are equatorial. This is because what the Natural Products Branch looks for is unique organisms that are in very rich environments, which requires them to compete with each other, because we feel that these organisms then are more likely to have to develop chemical defenses and unusual chemistry that allows them to compete with such an overabundance of biota. And so those are the collections we currently have. What you see here is pictures of our extraction laboratory. Bags of these extracts, whether they're plant or marine organisms, come to the NCI in Frederick, and they are ground and extracted in the Perkel columns that you see on the upper left. They are then dried down into these bottles that you see in the center using rotary evaporators that you see on the lower left-hand side of this slide. We then have lots of bins of vialed extracts that you see there in the upper right-hand corner, and then we take these vials and create plates for screening, 96-well plates, 384-well plates, that we then ship to researchers around the world to do research on cancer, and we provide them for free. The NCI provides these for free to researchers. All they have to do is pay for shipping. So, what exactly is in the repository? Well, here's a quick slide that shows you we have more than 200,000 different extracts in the NCI repository at this time. Actually, it's closer to 230,000 at this time. And you can see here the breakdown of them: about 160,000 plant extracts, about 40,000 marine extracts, and about 30,000 microbial extracts. These we have both aqueous and organic extracts for each source organism, and as I said, these are all plated out at this point and can be made available to researchers worldwide. So, what are the challenges? We now know that the NCI has produced a vast repository of extracts that should make available to researchers and should make screening extracts worthwhile endeavor and reduce a lot of the costs that might be associated with researchers doing this work. So what are the challenges? Well, for one, natural product extracts are difficult to screen in their crude form because they have cytotoxic compounds. They have common nuisance compounds that non-specifically bind to proteins. They also have fluorescent compounds and colored compounds. And so this makes working with some of the modern high-throughput screening systems challenging. And so researchers have to get some familiarity with how to work with screens with natural products. Also, each extract contains hundreds of compounds. And so once you get an extract that's active, you then have to go through the process of identifying the single active agent or family of agents that is in that extract. And that's a challenge for some researchers. And that type of purification and structural characterization of the active components, that's a unique skill set. And the time frames and the requirements for that haven't always meshed with modern high-throughput screening time frames. And I think that's part of the reason why we've seen not a lot of natural product extracts being used in the high-throughput screens. There's a requirement for a long-term, lower-throughput bioassay-guided fractionation support, and there's also a continual accrual of additional lead molecules that might not work in the time frames and such rapid screening paradigms that we use today. But I know there's a lot of words on this slide, so the easiest way to think about that is a picture is worth 1,000 words. Why is it difficult to screen natural products? Because they look like this. This is a plate of natural product extracts that are meant for screening. And you can see here, they're colored, they're sticky, they're viscous, they're fluorescent. And so having compounds and extracts like this put into screens is a challenge for many researchers. So what sort of decisions do you need to make when you're going to screen natural products? Well, what kind of density? Are you doing 96 well plates, 384 well plates, 1536? Most researchers now use 384 well plates for their screenings. But also recently we have completed a screen with natural product extracts in 1536, a higher density format. Also, there's the assay endpoint. There's common interfering compounds. You might imagine that a plant extract is going to have chlorophyll in it. Chlorophyll is green. If you have a green labeled compound that you're trying to find in your reporter assay, there's going to be complications with the green compounds that are present in those extracts. Also, extract concentration. There's several trade-offs here. You can screen at lower and lower concentrations of the extract, but then you're more and more likely to find common compounds that are only a high % of the total mass of an extract. So there's assay optimization that you can do to reduce these false positives. You also have to define your hit. Is your hit the same if you're screening marine extracts as it is if you're screening plant extracts, as it is if you're screening microbial extracts? Sometimes there can be significant differences in the type of molecules that are present in extracts from different sources. And so you might have to adjust your paradigms for screening based upon those source organisms. There's also hit prioritization, which is a requirement for this. It's high throughput, orthogonal, and it helps weed through some of these non-specific hits. So two things I want to talk about in more detail are assay optimization and hit prioritization to give you an example of why this is important. But the bottom line is that taking the time to address these decisions early on will result in a screening program that can effectively assay natural products and work better with the chemists and the downstream users of your data. So, assay optimization. What can you do to help? For example, you need to optimize assay kinetics in a cell-free assay system, and you have to optimize extract concentration. That means testing at a variety of concentrations, putting in a variety of enzymes. Also, as I mentioned, green labeled compounds can be challenging to use with natural products. So you could use a fluorophore or a label that has a longer wavelength shifted into the red region. This should help to remove some of the interference that you get from natural products. One important thing is just a simple wash step. If you can use a step where your label is bound to the plate and then you're able to wash away the unbound extract that's not interacting with your target, that allows you to wash away many of these interfering compounds. And finally, addition of something like an excipient protein like bovine serum albumin or gelatin to your assay buffer can reduce the effect of non-specific binding of certain compounds to proteins, because now the protein of interest is only a small percentage of the total protein in the well, and so that reduces the effect of those compounds on that protein of interest. And just to give you a visual example, here is an example of what an assay looked like before modification. And you can see that we have controls on the left-hand side of substrate. It's not inhibited. And then you see the presence of the enzyme there, TDP1, reduces the signal now down. And that window between those 2 points, the one on the left and the one in the middle, those 2 lines, that's your effective window of what might be a hit. What you can see on the right-hand side is a screen of actual compounds in this assay. So how many hits are in this assay? And you can see anything above that dashed line in the middle would be considered a positive result, something that is inhibiting that enzyme, in this case TDP1. That's an assay as it might come into the laboratory I work in, and it's not modified. This assay in our hands would be considered to have too high a hit rate. There's too many molecules that are becoming active here, and so we'd have a hard time weeding through them. So what we do is modify that assay using some of the techniques shown above, and this is what we get. And what you see here, now you have your controls, this time shown in red and blue in the same positions they were. They're tighter. There's a larger window between the two. And now if we take a look at the same exact compounds screened in this assay, now you see only a few compounds that have risen above that dashed line. That indicates that the assay is now significantly more rigorous. It's more challenging for something to inhibit that assay, and that's an assay that now might be more amenable to screening something as challenging as natural products. So, what other prioritization techniques can we use? There's a lot of things you can use here, and you have to really think down the line. You're still going to get a significant number of hits. If you screen all 200,000 extracts, you might still end up with 1,000 hits even after optimizing your assay. So you have to use additional assays, biological assays to determine cellular activity, gene transcript analysis, pathway analysis, or biochemical assays that look at direct modulation of your target. Can you get direct binding constants? Can you look for selectivity amongst related proteins with similar structures? And can you look at things such as reversibility of the inhibition and allosterism or non-competitive inhibition? Also, one could look at taxonomic and geographic. If you have a lot of hits from the same genus of plants, they might be one project instead of several. Chemical metabolomics can also help. This gives you an idea of the chemistry present in different extracts, and so some can be grouped and related by their chemistry rather than their taxonomy. And finally, there's just practical considerations. How much extract is there? Is it able to work in your separation techniques? Is it a very challenging extract or easy? So all of these ideas can be put to use in prioritizing your projects. Here I'm going to show you just an example of something we worked up on a project that we did with the Sanford Burnham Research Institute. It's a list of a variety of different criteria that we would like an active extract to have. And you can see there's some activity that fractionates well, there are things that are analytical as far as chemistry, and there's also taxonomic considerations. And every extract that came out as a hit got points associated with being able to pass certain of these criteria. We're then able to grade the extracts and come up with a triage triangle like you see here. So there were over 3,000 initial extracts that hit, but you can see after we did confirmation and validated them through certain techniques, and then we ended up with only 100 that were fractionation candidates. By the time we got down to structural elucidation candidates, we only had 22 extracts, which is a number that's easily doable by most chemistry labs. And this then yielded a couple of interesting molecules that might be useful for drug development. And that's the type of triage that you have to do to be able to weed through all of the natural products. So I'm just going to go over a couple details on a couple different assays that we've done recently to give you examples of the type of screens done in the NCI. And in this case, we're going to talk about MALT1, which is an enzyme, and also TDP1, a different enzyme that were done in our lab, and they're different types of formats. These are just a list that you see here, a variety of assays that have been done in the Molecular Targets Laboratory in which I work. So the first project is a MALT1, and this is involved with diffuse large B-cell lymphomas. So these are 40% of non-Hodgkin's lymphoma, and there are about 23 diagnoses per year. Unfortunately, this only has a 40% cure rate and approximately 10,000 deaths per year. So there was a genetic analysis done by Lou Stout's laboratory in the CCR that was able to find different genotypes of lymphomas to separate them based on the actual genetic and proteinaceous differences between these types of lymphomas to get a better idea how they could be targeted. And what he found was that an activated B-cell-like diffuse large B-cell lymphomas were the most pernicious form. They had the least survival rates. They were the most challenging to treat. And so he came to us looking for ways that we might be able to find molecules that target these particular types of lymphoma. And so one of the aspects that he found was that NF-kappa B was found to be constitutively active in these activated B-cell lymphomas. So this shows some of the key pathways, and I know it's a very complex diagram here, so I'm going to home in on just some areas right in the center here. And what you see there are a variety of molecules, in this case proteins, CARD11, MALT1, BCL10, and you can see a little box there called MALT inhibitors. And what we're looking for are things, if you look below, these things all turn on NF-kappa B pathway. And what we want to do is turn that off in these lymphomas. And so what we did is look for which molecules we might be able to inhibit. And it turned out that MALT1 is an enzyme, a protease, and that was what we targeted. So we set up an assay for inhibitors of MALT1, which is a translocation protein. And it's active in these diffuse large B-cell lymphomas. And it's a hallmark of them, so that the MALT1 is also an interesting protein in itself. It's a paracapsase, which is unusual in the human genome, in that it is an arginine-specific protease, and there aren't many of those, so that made it potentially interesting. Also, Dr. Stout's lab used siRNA and peptide inhibitors of this protein and showed they were selectively toxic to MALT1-dependent diffuse large cell B lymphomas. So, this is the protein you see here is the paracapsase, that is MALT1, and what we set up was an assay to identify small molecules. So this is a substrate for MALT1. It has 4 amino acids and it has a label on the right-hand side here. MALT1 will cleave that amino bond that you see there, that amide bond, and it will free that fluorescent label. So what we did is we set up an assay that could cleave that label off of it, and that would show us the activity of MALT1. If we added extracts to that, they would inhibit MALT1 and they would increase the fluorescence of that substrate because MALT1 had been inhibited. So, first thing you have to do for an assay of this type is you have to optimize the kinetics and the biochemistry of the enzyme. So we looked at a variety of different forms of the enzyme. We looked at the time scale of the reaction and the velocity of that reaction. And you can see here that GST MALT1 had about 100 micromolar km. So that seemed a good level of enzyme kinetics and turnover rate for us to use for the assay. You can also see that we selected a time point of 60 minutes, which was right in the middle of that velocity curve. So there's still linear velocity out to 120 minutes. So if we ran the assay for 60 minutes, there would be a constant rate of enzyme activity. And that's important to give you optimal use of an assay. And what you see on the lower right-hand side is an assessment of assay repeatability. The common term that is used is a Z factor. And a Z factor should be above 0.5 to be useful for screening. And it tests multiple plates on multiple days at multiple times. So each plate has 384 wells in it, and each well is an individual assay. And you repeat that multiple times and show that it has significant repeatability. You get the same results day in, day out, no plate differences. And you can see here that our averages were well above 0.5 in all cases, indicating that this assay was useful for screening and robust enough to do natural product research. Here, like the triangle I showed you before, gives you an idea of what our throughput was. We screened 108,000 extract samples. We found 879 that were originally appeared to be active. Again, we can confirm those in quadruplicate. We did secondary assays for selectivity and reversibility, and then we had selective cytotoxicity in a cell-based assay. So our primary assay was biochemical, looking at just inhibition of the enzyme. That secondary assay was in whole cells, looking for selective killing of MALT1 cells. We ended up with 37 projects that were worthwhile for chemistry. This just gives an example of a few of the ways we looked at selectivity or specificity. We tested against 2 other enzymes, ARG-C, which is another arginine-specific protease, and also caspase-9, which is similar in structure to MALT1. And we took a look to see if any of these molecules were selective. And what you see here are the inhibition constants in micromolar for the different molecules we found. And what I'll show you here is how we did extracts. With extracts in the cell-based assay, we were actually looking for changes in an NF-kappa B specific reporter. So we know we have a molecule that inhibits the enzyme of interest. It shows selectivity, and now we want to see in cells, does it selectively downregulate NF-kappa B, which is really the endpoint we want to be able to attack these lymphoma cells. And what you can see here in the graph, that we want to see the positive control, in this case ibrutinib, you can see that the left-hand side of those graphs move in an upward direction, going from blue to green to purple. And on the right-hand side, you can see they go down in the opposite direction. That is the positive control. That's the signal we're looking for in extracts. We tested 188 extracts, and 37 showed activity like that, where that first one was going up and the second one was flat or going down. And this is an example of what one of those positive extracts would look like. We then took a look at this particular extract, which was a marine extract, and we saw that it had activity, again making the luciferase go up and leaving the NF-kappa B flat. We saw that it inhibited the enzyme, and it turned out that we were able to isolate these molecules that you see here, the calabugilones. They were isoquinoline quinones, and they were isolated originally from a bryozoan, a marine organism. They showed activity against MALT1 at a 15 micromolar concentration, and that was not affected by glutathione. They showed selectivity, a little better selectivity for caspase 9. But then we took a look at this molecule, which is a synthetic derivative of the calabugulones, and you can see here in cell-based assays. What we're looking for is something at the top that is not affecting growth, and in the bottom graph that is, the opposite way. In the top is affecting growth, but in the bottom is not. And so what you can see here, the top graph shows activated B-cell lymphomas. The bottom graph shows germinal B-cell lymphomas. So that's the difference. The top has NF-kappa B active, and that's what we want to inhibit. The bottom graph doesn't. So we want to see activity up top and not at the bottom. And what you can see here is that compound I showed on the previous screen selectively kills the activated B-cell lymphoma cells, but does not touch at 10 micromolar the other cells. And that's the type of selectivity we want. And so that molecule is moving forward for further study and potential modifications to increase its potency. Now I'd like to move on to a different type of assay. This is actually looking at an assay that attempts to restore sensitivity to topoisomerase inhibitors. And this was a collaboration we did with Yves Palmier in the developmental therapeutics branch. So, what is topoisomerase? Topoisomerase is an enzyme involved in DNA replication and repair. It's an important enzyme. Your cells can't grow without it. However, cells that are rapidly turning over, like cancer cells, are more sensitive than topoisomerase inhibitors than others. And in fact, there are known natural products, such as camptothecin, which are inhibitors of topoisomerase. They've been used in the clinic for years. They're very effective. And what you see here in the bottom lower right-hand side of this is that you see topoisomerase comes, it makes a cut in DNA, and then it allows it to unwind and then repairs it. What happens is when you add camptothecin into this, camptothecin binds and prevents the topoisomerase from being released. And so you build up these perturbations to the DNA, and the cell cannot tolerate that. So after a period of time, the cell dies. However, there are mechanisms for recovery from these type of injuries. Over time in evolution, cells had to adapt to be able to handle this type of perturbation. And so what you see in the next is there's an enzyme called Tyrosyl-DNA phosphodiesterase 1, or TDP1. Now what TDP1 does is hydrolyze that covalent bond to topoisomerase. And so when camptothecin blocks topoisomerase and freezes it on the DNA, TDP1 can come and cut that off. And that allows cells to become more resistant to topoisomerase inhibitors like the natural product camptothecin. So what we wanted to do was develop an assay to look for inhibitors of that enzyme. To inhibit TDP1, then maybe we restore sensitivity to topoisomerase inhibitors. And so, as you can see here, we replaced topoisomerase 1, this time with a different fluorophore, a redshifted fluorophore, so we could see the activity of this enzyme. And as I showed you previously, we were able to optimize the enzyme kinetics and all the parameters of that assay to have a very robust assay. And this just shows some of the statistics for that assay. We screened synthetic compounds, partially purified natural products, natural product extracts, and you can see overall we screened over 200,000 different samples and had a total number of hits of about only 117. So this was a very rigorous assay. What you can see here is a secondary assay that was done. Even though we had only a low number of confirmed hits, they had to be confirmed. In this assay, instead of looking for changes in color or fluorescence, we're looking for changes in a gel shift assay for the presence or absence of a band that shows that TDP1 was able to cut. And what you can see here, for example, in this marine extract, C026531, you can see that the bands are missing at the higher concentration shown by the fat side of the triangle above the gel bands. What you can see there is that in the first 3 highest doses, there's no lower band, indicating that TDP1 is unable to cut that piece of DNA, meaning that it only goes at the higher molecular weight and there's no lower molecular weight band. And you can see as we go to lower concentrations that yes, we are able to see that lower band because we get below an effective therapeutic concentration. So what are the compounds that we were able to isolate? Here we found additional marine compounds. this time from a sponge called Zestaspongia. There was a family of compounds, the Halenaquinols, that were isolated from this. They were confirmed in the gel shift assay. They also were tested to increase the presence in cells of topoisomerase adducts, which they did, and they did not inhibit they selectively inhibited TDP1, but not a similar enzyme called TDP2. And so these look like potentially interesting molecules, but how do they work? Well, one of the things you can do is look at the biochemistry of how these work. What you see on the upper left-hand side is a graph that shows changes in the velocity of enzyme activity after the enzyme has been treated with drugs, but the drugs have been washed away. So you pre-treat the enzyme with drugs, you then dilute the drugs out to well below any effective concentrations, and then you look at enzyme activity and see if the enzyme can still be active. And what you see in the circled areas and in those lines that are down near the bottom of the graph is that those are indicative of irreversible inhibition. So that once we treat the protein with the drug, even though we wash the drug away, it's still stuck to that protein, still stuck to TDP1, and still able to inhibit its activity. And that was the case with these drugs. So dilution of halenaquinone sulfate away from TDP1 did not reduce its inhibition activity, indicating that it was likely covalently bound to the protein, a suicide inhibitor, if you will. The other aspect we looked at was what type of inhibition. TDP1 normally uses a certain substrate. It's going to bind to DNA, but it also uses ATP. Now, one of the things you look for in enzyme inhibition, if you are competing with the substrate for binding to the same site the DNA does, that would be considered competitive inhibition. What we're looking at are things that are going to be non-competitive. So they don't bind to the same site as the DNA. You don't necessarily want a DNA mimetic. You want something that acts at a different point of the enzyme. And what you can see here from the lower graph on the left-hand side is classic Michaelis-Menten kinetics indicating non-competitive inhibition for halenaquinone sulfate, indicating that it's binding somewhere other than the substrate binding site. And that's important because that means we might be able to identify new biochemistry and new biology about this protein and find some new site that drugs could be able to inhibit. So, halenaquinone itself is an interesting molecule. It could react to covalently bind two different ways, one binding to cysteine residues and one binding to lysine residues. So which way were we seeing this react? That was one of the things we wanted to look at. Well, it turns out, in fact, that halenaquinone sulfate, which only has area 2 here that you see, the lysine preference possible, because area 1 is no longer quinone. It is sulfated at that position and so is unable to react with cysteines. So we knew that this molecule and the mechanism by which it was inhibiting TDP1 had to go through an adduct onto lysine rather than cysteine. So if we take a look, there's already been a drug in clinical trials, phase 2 clinical trials, called wortmannin, or an analog of wortmannin, which you see on the upper left-hand side here. And wortmannin has a very similar structure if you look to halenaquinone sulfate, especially in the bottom part of it that reacts with lysine. And so we wanted to take a look to see if wortmannin would work in a similar mechanism as halenaquinone sulfate against TDP1. What we found is that wortmannin was inactive against TDP1, which indicated that the other parts of this molecule outside of that red circle must be important for the selectivity of halenaquinone inhibition against TDP1. So what is the structural basis for the inhibition? What part of TDP1 has a pocket that will fit halenaquinone but won't fit wortmannin? And how does that work? To get that answer, we were able to do structural studies, and what you see here is the three-dimensional structure of TDP1. We were able to get crystals. A colleague of mine, Tanush Mulwe, who worked in our laboratory, was able to get co-crystals of TDP1 with halenaquinone sulfate. Now, TDP1 itself has 37 different lysines, so there are 37 different possibilities that halenaquinone sulfate could bind. What we found, however, was that this region that you see here highlighted is an unstructured loop that was found in the back of TDP1, away from the substrate binding site. And when we took a look at where TDP1 was bound, we found that it's selectively bound into that site with those 3 lysines. And if we look in more detail in the preliminary X-ray crystal data, there was a particular lysine, lysine number 556, that halenaquinone sulfate bound to. When we used mass spectrometry to identify if there were any other lysines that were modified by TDP1, we did not find any other lysines. Only a single lysine in TDP1 was modified by halenaquinone sulfate, indicating that this region was sufficient to inhibit the enzyme, even though it's 180 degrees away from the binding site for substrates. So that gives us important information of additional molecules that might be able to be designed to bind into this site to be more drug-like and inhibit TDP1. You can see here some of the points of that interaction. So I've shown you some examples of how to screen natural products and the ways we do that and some of the challenges. So how can we make this better? You know, a significant cost in time and resources is the effort to do the natural products isolation and structural elucidation. So here you're going from a crude extract through fractionation in a column like you see here to the characterization of an active molecule. You can see here an example of some of the chemists that might be working in your laboratory doing this. So how do we get natural products to be a little more efficient for us? Well, one of the ways is to pre-fractionate the extracts themselves. So if we partially purify the extracts, what effect will that have? Well, what we see is that in cell-based assays, we see an increased hit rate for those enzymes, indicating that we might have concentrated minor components and increased the percent of the active compounds in those fractions. In cell-free assays, like the ones I just showed you, for example, TDP1, we see lower initial hit rates, meaning that we have purified away some of those nuisance compounds that gave us non-specific activity. And so we get a higher percentage of hits that confirm, because those extracts that show activity, or fractions that show activity, now have a higher rate of confirmation because they're freer from those nuisance compounds. So prefractionation removes some of the non-specific false positives and also increases that reconfirmation rate. So what the NCI has decided to do is they're undertaking a large automated prefractionation of natural product extracts that's going on right now. And what there are plans to do is create a 1 million fraction library of semi-pure natural products for screening. We hope that this will help sequester nuisance compounds and concentrate these low percentage active metabolites. And this will provide a value added, as we'd like to call it, screening library, unlike anything currently available. And like the crude extracts themselves, this will be made available to researchers worldwide for free. And then the use of this prefractionated library should increase the efficiency of both high throughput screening and subsequent chemistry efforts. Just to show you an example, we're also undertaking rapid secondary analysis. So in this case, we have prefractionation of a crude extract, and you see the extract up at the top, and it shows activity. The farther those bars progress to the right of those graphs, the more active the component in that fraction. You can see that that activity was concentrated in just fractions 4 or 5 here. Fractions 1, 2, 3, 6, and 7, the graph bars are much shorter, indicating there's not much activity there. Whereas fractions 4 and 5, the bars in those graphs are longer, indicating more potent activity in those fractions. So what we now undertake is a rapid secondary analysis, and we've built high throughput mechanisms for this so that we can create 1,000 subfractions per day. We can basically take that initial fraction that showed activity and further purify it into 22 subfractions. These fractions can be done in an automated manner where we can do 44 fractions in 13 hours on one instrument. And what we can then see are standardized chromatography runs, like you see below, separating these compounds based upon their lipophilicity, or C log P. So what will this allow us? Well, rapid prioritization of hit lists. So now if you get 100 or 200 things, we've set up a system whereby you can screen 400 of these through the second level purification and get those plates back and have that complete within 2 weeks. This should improve the speed and efficiency of hit confirmation by screening laboratories, and it will assist both intramural and extramural researchers in the identification of new active natural products. And also, an important point here, although it's the last one listed here, is this will minimize the waste of the extracts. The Natural Products Repository is a national treasure. It's unusual anywhere in the world. It is something that is not being recreated. Some of these extracts are the only ones we have of those, so finding the chemical diversity in them is important. By using this methodology, we use much less of the extracts, which preserves that repository for future generations and future researchers. And so that's very important as well. So one of the ways we can look at data is important too. So now with bioinformatics, we can look at large swaths of data. And with the new prefractionated library being large and 230,000 extracts to start, there's a lot of data to look at. And what you see here is something we call a self-organizing map that members of our laboratory created to evaluate the biological activity of the extracts in aggregate. So we look at all the extracts at once. In this case, we're looking at their activity in the NCI 60 cell line panel. So that is a panel of 60 different human tumor cell lines. So each of the extracts was tested in 5 different doses against all 60 cell lines. So that gives you 300 data points for each extract. And what the self-organizing map does is look at the pattern of activity. Basically, it's like a pattern of dots of the activity against those 60 cell lines, and they compare that to the pattern of dots from every other cell line. And they keep comparing them, and it's self-learning. So what it does is it keeps looking and looking for matches and closer, and when it finds things that are close enough, they go in individual nodes, which are the circles you see on the graph here. Each one of those circles has approximately, 10 or so extracts in it, indicating that their activity in the 60 cell panel, the pattern of that was close enough that they might be related. And so what we do is we think this puts those extracts into biological space, which is basically our way in this assay of looking at their activity against cancer cells. And what we do is we parse this self-organizing map by increased lethal response or by increased potency or lower initial concentration of response. Now when you look at it in aggregate, you get a huge map like this. This is looking at every extract that's ever been tested in the NCI 60 panel and every pure compound that's ever been tested in the NCI 60 panel. And what you see here are individual nodes now that contain about 25 compounds. Those nodes that are lighter in color than the other nodes are nodes that only contain extracts. There are no pure compounds in those nodes, indicating that maybe that region of biological space might be a good area to look for new chemistry. And what you can see here are numbers on a few of these, and those numbers are particular extracts that might be of interest. This is an example of something we worked on. So we looked at 26 pure compounds through this, and we're able in a short period of time to identify 26 pure compounds, 2 new natural products, and do that in a period of only 6 months. One of the aspects in looking at natural products are these going to be drugs? And so what we did is we wanted to look at the chemical space coverage of these. And this basically takes a look at different parameters that are involved in chemistry. The weight of something, its lipophilicity, its aromaticity, perhaps its flexibility by sp3 carbons. What you see here is all those 26 molecules that we isolated on a 3-dimensional graph of lipophilicity, molecular weight, and aromaticity. And you can see they're nicely spread amongst those parameters. Here we've exchanged the x-axis for flexibility this time while retaining aromaticity and lipophilicity. And what you can see is, again, we have a nice spread of compounds with a nice amount of sp3 carbons, indicative of what is typical for natural products. Now, importantly, what we wanted to do is compare what these look like as drugs. And so there were methodologies that are used to compare compounds to each other. In this case, we wanted to compare them to all approved natural product drugs. And what you see here, we took natural products approved from 1981 to 2010 and compared them by the same parameters we just compared those molecules that we isolated using a statistical program, ChemGPS. And what you can see in this next graph is you see in 3 dimensions again, natural product drugs, aromaticity, molecular weight, and lipophilicity. The natural product drugs are shown in blue. The natural products that we isolated with our test system for this new high throughput methodology are shown in orange. What you can see is there's a significant overlap in the chemical space in this case. Instead of the biological space we looked for with the SOM, Now we're looking at chemical space, and what you can see is a significant overlap of known drugs with the type of compounds we're finding through these methodologies. The only area that's missing is you can see this area down here, and that is an area that is a much more polar compounds. Those don't seem to be present, but that is likely because our test set was only organic extracts, not aqueous extracts. And aqueous extracts are going to be more likely to contain the type of polar compounds that you see down in that lower region. So as we get more data in the future on both aqueous and organic extracts, we are hopeful that we will have continual overlap with what is required from natural product drugs, so that we know we're identifying the type of molecules that could lead to clinically useful drugs. So with that, I would just like to end and basically let you know that this process of prefractionation with self-organizing map analysis, metabolomics, rapid isolation and structural elucidation will come up with pure natural products that are useful. And I hope some take-home messages are that you will see that natural products continue to be an important source of chemotypes resulting in approved drugs, that screening new crude natural product extracts requires assay modifications, and hopefully you've learned a few of those today, and that the high hit rates with extracts require you to use different prioritization techniques to be able to weed through those extracts and find those that could be most useful. Also, prefractionation can make a significant difference in new automation technologies in the throughput and efficiency of natural product efforts. And finally, these new bioinformatic interfaces will allow sort of an omic level of prioritization and evaluation of natural product research, and hopefully improve natural product discovery. And with that, I just want to acknowledge several people in different laboratories who contributed to the data I showed you today. So with that, I would like to thank you very much for your attention. I hope that I've been able to show you some new aspects of natural product research, and hopefully some of what you've seen today will encourage some of you to get involved with natural product research, because there's a lot more things to find out there, and hopefully we'll find more molecules that will be helpful to the cancer patients we try and treat here at the NCI.